Since roots are directly responsible for soil resource capture, our group is particularly interested in genetic variation for root anatomy that may affect how efficiently different crop varieties acquire and utilize resources like water, nitrogen, and phosphorus. To collect samples for anatomical analysis, roots must first be excavated and washed in the field. Here we have a fresh maize root crown that was just excavated from the field, and we've taken it back to the lab to collect root samples for anatomical analysis. At first glance, this maize root crown may look like one big tangled mess of roots, but upon closer examination, we can see how the maize root system is organized into distinct root classes. Here we can see the mesocotyl, where the seed initially existed, and the primary root which emerged from that germinating seed. The rest of the subterranean roots you see in this crown are referred to as nodal roots, and upon closer examination, we can see that they are organized into distinct rings or whorls. Here we see the first whorl of nodal roots. These are the oldest nodal roots in the crown, and are the first nodals to emerge after germination. As the plant goes through its vegetative stage of growth, new, subsequently thicker nodal roots are emerging above the older roots in the crown. So here's the second whorl, third whorl, fourth whorl, and fifth whorl of nodal roots. These pigmented roots, which are emerging above the soil surface, are called brace roots. Because these nodal roots are developing over time while stresses like drought may be becoming more severe through plant growth, our group is interested in collecting anatomy samples from across these different whorls to look at how anatomy may change from whorl to whorl. A sharp set of pruners are an ideal tool for removing roots from the crown. It is important to be consistent in sampling location from crown to crown, and we typically focus on the basalmost 2 cm of each nodal root, right where they emerge from the crown. Anatomy samples collected will be stored in 2 ml tubes filled with 75% ethanol. This will preserve the root samples until we can image the anatomy of these roots using our laser ablation tomography system at a later date. Using our laser system, no additional prep work to fix or stain samples will be needed, and they can be sectioned directly from storage in the ethanol solution. Genetic variation in the anatomy of these maize roots may affect the metabolic efficiency of the roots in foraging soil resources, the ability of the roots to penetrate strong soils, or the transport of soil resources through the roots to the shoot. Anatomical features of interest from these samples include the size and abundance of cortical cells, xylem vessels, and air pockets in the cortex, known as parenchyma.